Hi friends. So today we are going to see a widely used technique called PCR. So what is PCR? It is polymerase chain reaction. This polymerase chain reaction. So why it is called polymerase chain reaction? So a product from first step of reaction, if it is helping in additional reactions and if it is accumulating exponentially, then we will call that as a chain reaction. So in the case of PCR, a product DNA fragment is forming from a first step of reaction, and in the additional reactions it is getting accumulated with the help of a enzyme that is called a DNA polymerase, and that is why it is called, because a polymerase enzyme is involved, that is why it is called a polymerase chain reaction. Otherwise the chain reaction is enhanced by a polymerase. So this polymerase chain reaction is also called in vitro cloning. Why it is called in vitro cloning before PCR is coming? Identical copies of a subject or DNA is made by cloning. That is cloning, that is gene cloning. Cloning in a plasmid vector or lambda vector or yak vector or back vector. And the desired copy fragment of DNA, the copy number was increased by cloning. So that is called in vivo cloning. So polymerase chain reaction is called in vitro cloning because there is no biological material is involved. It is doing it in a tube. That is why it is called in vitro cloning. So in a PCR, we are making identical copies of a particular DNA sequence in large number using chemical reaction mediated by a polymerase enzyme in a tube, in a plastic tube. That is why it is called in vitro cloning. So before this PCR is coming, we were making a double-stranded DNA by chemical synthesis. First, all of you know that a DNA is double-stranded. So both complementary strands, that is plus strand and minus strand, were chemically synthesized. Then they were renatured using renaturing mechanism, and we were making the double-stranded DNA. So this is actually very cumbersome process. Lot of time is involved. So because of that, people were looking for a very easy method, which we can do it in a very short period of time. And in the chemical synthesis, the repeatability was also very difficult. So that is why. So this is a method actually proposed by Karry Mullis and his colleagues. So they were actually looking for an enzymatic method so that this could be done in a very easy manner. So invention of PCR that made the synthesis of double-stranded DNA very easy. So what are the discoveries which helped in the PCR technology development? As you know, Watson and Crick, they discovered that DNA is double-stranded. So Arthur Kornberg, he purified an enzyme called DNA polymerase, which is responsible for DNA replication mechanism. And Karry Mullis told that if you are using a primer, an oligonucleotide, with the help of a polymerase, you can make a double-stranded DNA. So initially they used DNA polymerase. So later the process was very easy or became very easy by isolation and purification of a thermostable DNA polymerase from Thermus aquaticus. So Frederick Sanger, he used this method, that is what Karry Mullis proposed, a primer and a thermostable DNA polymerase. So he actually used that and with the help of DNDP. So he used that concept to make DNA sequencing or to perform DNA sequencing. So it was in 1983, Karry Mullis, he used the DNA sequencing method as a basis to devise a new technique. So he added a second primer. So when Frederick Sanger was doing, even though he was doing a PCR technique, he was using only a single primer. So he was not getting DNA in an exponential manner or large quantity. The yield was not getting in a large manner. So in 1983, Karry Mullis, he introduced a second primer also. or you can say Karry Sanger was using only forward primer. So Karry Mullis he introduced a reverse primer also. So he could amplify or he could amplify the DNA in a very exponential manner. That is how he discovered, he achieved the product formation in exponential manner. So that is why he got a Nobel Prize also. So the repeatability also became very easy. And so initially Karry Mullis was using DNA polymerase. So it was a very cumbersome process because as you know, so these are the PCR machines which initially used. So this is actually a pipetting device. It is not a PCR machine like we are using. It is a pipetting machine, and this is also a pipetting machine only, but it is a little bit automated. So in Karry Mullis, when he was doing the PCR, so after you know a little bit about the PCR, that is I am telling you. So after each extension step, when you are denaturing the DNA into single strand, the DNA polymerase was getting inactivated. So in each step, you have to add fresh amount of DNA polymerase for the next step. So that was a very cumbersome process. So in this method, people used the thermostable enzyme also, that is Taq polymerase. So when the Taq polymerase was introduced, you saved, that is, you don't have to add a fresh enzyme for each step. So in later, they also introduced a computer algorithm into the PCR machine, and also Peltier effect into the PCR machine, and they could automate the PCR cycle as we are doing now. So, so these are the different PCR machines currently earlier we used and currently in use. So earlier when you see the PCR machine, these are all single block PCR machine. So now you have a multi-block PCR machine, and even in a single block you can set a different annealing temperature. That means if you have a PCR block of 96 wells, so you can divide, you can have a block of 16. So one block will be having around 16 wells. So like that you can have several blocks. So you can standardize the PCR very easily. Otherwise, in the earlier PCR machine, because it was a single block, so when you, if you want to do a annealing temperature standardization, a gradient was set, and you don't know when a product is forming in a particular well, you don't know what is the temperature maintaining there. So you have to use thermometer or some other techniques you have to measure the temperature what it is maintained. But it is it will not be accurate because when you open the lid, the temperature there will be slight change will be there. So because of that, it may not be accurate. So you can set in each block what is the temperature you want. You can have a temperature range. Suppose if you are 2-degree difference, 1 to 0.1 to 2-degree difference you can maintain between these two blocks. So like that you can make it. So like that they have now there are PCR machine available which you can use. So when you say a PCR, there are components like template, PCR buffer, then primer, DNTPs, polymerase, MgCl2, and water. These are the main components which are normally when you use when you are setting up a PCR reaction. And in some cases, you may use glycerol and DMS also. Suppose if there are any difficult to amplify template is there, because if it is, there is a highly GC rich template if you are using. So in such cases, or when there is a hairpin formation is there in the PCR product. So in such cases and all, people will use glycerol and DMSO and try to get a product in large quantity. So otherwise, in a normal PCR, you can use template, buffer, primer, DNTP, polymerase, MgCl2 and water. Water is mainly used for making to dilute the PCR buffer to 1x, because in most of the cases buffer will be supplied as 10x. So that you can mix template, primer, DNDPs, polymerase, MgCl2, everything, and finally when you add water, the buffer concentration will become 1x, which is actually a reaction concentration. So generally template what you are using is either an isolated genomic DNA or a PCR product or a plasmid or some forensic samples or the RNA. So these are the template which are generally used in a PCR reaction. So buffer usually contain tris base, potassium chloride, EDTA, and like that. In some cases, when you purchase a buffer, this MgCl2 also will be substituted in this. So you can purchase PCR buffer without MgCl2 and with MgCl2. So the primer, if you say these are oligonucleotides of around 18 to 35 base oligonucleotide. Generally, a oligonucleotide of 18 to 21 is preferred for PCR reaction. So there will be a forward primer will be there, and there will be a reverse primer will be there. So in the case of DNDPs, so the DNDPs are the building blocks of the double stranded DNA. So there you can use either free DNDPs or labeled DNDPs or unlabeled DNDPs or conjugated DNDPs, depending on the purpose if you want to do. Suppose if you are using any probe preparation, you will be using labeled DNDPs. That is, DNDP will be labeled with any of the radioactive material, or it may be conjugated with some of the fluorescent or quenching units. So that is why you can use it. So depending on the DNDPs what you are using for the PCR reaction, you may slightly take a decision what polymerase to be taken. So this MgCl2 is actually helping in the stringency of the reaction. Suppose if before adding glycerol and DMSO, you can slightly change the concentration of the MgCl2 and you can get a good amplification. So when you select a polymerase, so the polymerase is actually you have to see thermal stability, extension rate, fidelity, and processivity. So this you will consider of a tagged DNA polymerase. You will consider for what type of PCR product to be formed, or how long the cycle will continue like that. So thermal stability means you are actually, when you do a PCR reaction, you are changing the temperature 95 to 55 to 72, 68. So like that, there are a lot of temperature changes there. So even after 30 cycles, the enzyme should show at least 90% efficiency. Extension rate means what is the length of the PCR product can be made by the track polymerase, or in a second how much nucleotide can be added. So generally extension rate is told like 1000 base per minute. So fidelity means it is accuracy of adding the nucleotide. So when the PCR is continuing, or when the polymerase reaction is continuing, whether a right nucleotide is added in the PCR product. Processivity means how long the enzyme and the template is bound before it is leaving the template. That is called the processivity. So there are a lot of polymerase enzyme used. So DNA polymerase was the one which initially used. Then after that, so this polymerase also, when you take, it has three types of activity. One is 5' to 3' polymerase activity. Yeah. 5' to 3' polymerase activity, and 3' to 5' exonuclease activity, and like that. So some this DNA polymerase also have 5' to 3' exonuclease activity. So this Clino, they have done is they have removed the 3' to 5' exonuclease activity, so that you can use either labeled or conjugated or like that. So when you use, the fidelity will be slightly changed. So you can also use T4 DNA polymerase, Taq DNA polymerase, terminal transferase, and some of these polymerase also have terminal transferase activity. When you take a Taq polymerase, it will always add a nucleotide at the 3' end. So this 3' end nucleotide will help you in the TA cloning when you get a PCR product. So this PFU DNA polymerase is having 3' to 5' exonuclease activity also. So if you need a very high fidelity, if you are expecting very high fidelity, you can use a PFU polymerase. Sequenase is an Taq polymerase, which is a mutated Taq DNA polymerase, which is generally used for Sanger's DNA sequencing, because you are using a labeled DNTP. So it often, when a normal Taq polymerase you use, you won't get a long extension. So when you use this sequenase enzyme, you will get a very good amplification, or the product length will be more. Length will be large. So you can also use deep went, Hercules enhanced fusion, ART DNA polymerase, BST DNA polymerase. So this RBST DNA polymerase is the one which is used in the isothermal amplifications. Means you can do PCR amplification at 32 or 37 degree room temperature you can do. So this BST DNA polymerase. So like that you have a lot of variations. So you can go through the enzyme properties, and you can use whether you need a high fidelity or you need a long PCR product to be formed, or like that you can decide what DNA polymerase to be used in the PCR reaction. So this is typically a polymerase chain reaction look like. So if you see, there is a first step is there, which is initial denaturation at 90. So the template is template along with the primer, buffer, Taq polymerase, MgCl2, water. This is actually boiled to 95 degrees centigrade. So usually a PCR buffer will be at room temperature, it will be alkaline. So it will help in the denaturation very easily at 95 degrees centigrade. So later, so you also know that the pH is dependent on the temperature. So when the PCR buffer cools down to the extension temperature, it will be an ideal pH for the enzyme to perform. So you don't have to worry about it. So initially the initial denaturation is 95 degrees centigrade. So next is the one which is actually a PCR cycle. So there is a 3-step is there. One is denaturation, annealing, and extension. So this initial denaturation is used because in the first template what you have used will be mostly a longer template when you use a genomic DNA. So this usually, if you are using a genome DNA, you have to incubate for 1 minute. Suppose if you are using a very short template like PCR product, you don't have to keep for 1 minute, you can keep even 30 seconds. So this similarly, so when you come to this cycle, so after the first cycle is formed, this denaturation, you can keep it like 1 minute to 30 seconds to 1 minute. So depending on the product size. So this is also 95 degrees centigrade. Then you are cooling the PCR tube to 37 to 68 degrees centigrade. So 37 to 68, what it's a range actually. So this temperature you have to decide based on the TM of the primer what you are using. So suppose if you are finding that you are using very short primers and all, you cannot keep at 68 degrees centigrade. So you have to incubate at 37. So depending on the TM of the primer, you can decide what is the annealing temperature to be set. So this annealing also generally people will do from 30 seconds to 1 minute. So this also, if you are using a very long primer or high complementarity is there between the template and the primer, then you can keep it for 30 seconds. So otherwise, if you are thinking that there are some mismatches are there, then you have to keep for 1 minute. So extension temperature is again 68 to 72 degrees centigrade. Usually tag polymerase, when you are using, the extension temperature will be 72 degrees centigrade. And when you are using this high fidelity DNA polymerase, then you have to slightly reduce the temperature to 68 to 72 degrees centigrade, because this also has a 3 prime to 5 prime exonuclease activity. So because of that, if a long product to be formed, you have to incubate from 68 to 72 degrees centigrade. Now commercially there is some mix is also available, where they have mixed the tag polymerase and the high fidelity DNA polymerase in a particular ratio to get a high fidelity long PCR products. Next step is the final extension. So this final extension, why is the, people are or we are following it is, suppose when this is the template, and when the PCR product is formed, you may expect that the PCR product should be formed up to this place. But most of the time what will happen is, when the actual PCR reaction, it may extend up to here only. So this much portion will be unfinished. So this unfinished product can be later polished to its original length by setting up a final extension. So this final extension time, suppose extension, if you are keeping 1 minute, then final extension will be around 5 to 10 minutes. So like that you can keep it. So this 1 minute also, how this extension time you are deciding is, so suppose if your PCR product is around 1000 base pair, then you have to keep 1 minute, because you are assuming that the extension rate is 1000 bases per minute. So that is why you are keeping 1 minute for 1000 base pair product. So like that. Then after the final extension is over, you are cooling the PCR product to 4 degrees centigrade. So this you can keep it for either 10 minutes, or you can keep it infinity. But infinity is not normally not recommended, because it may damage the PCR wells because of condensation of water. So if you see, there are step 1 is there, 2, 3, 4, 5, 6 steps are there. So from step 2 to step 2, 3, 4, it is actually a looped process. So this will continue around for 30 to 40 cycles. That is how PCR product is accumulating. So this is actually the reaction what is going inside the PCR tube. So initially you are putting one copy of a double stranded DNA. So this double stranded DNA, when you are denaturing at 95 degrees centigrade, so it is becoming single stranded. So when you are doing annealing, so these are the primers. So these primers will anneal, or it will base pair with the template. It will base pair with the template, and when you are doing the extension, these primers will add DNTPs to the 3' end, and it will form the PCR product, and it will become double stranded. And in the second cycle, so in the second cycle, what it will happen is, in the denaturation step, these are again becoming single stranded. Then in the annealing step, the primers will bind, and the PCR product will be formed from the original template and also from the product of the first cycle. So after the second cycle, you have 4 copies of the DNA. But if you see, the specific product is formed, what is formed in the second cycle will be further amplified in a logarithmic manner. So generally what we will say is, the number of PCR product formed will be 2 raised to n copy. N is actually the number of cycles what you are setting in the PCR process. So, but the actual number formed will be 2 raised to n minus 1 copy, because the specific product is formed only after the first, only after the second reaction. So from the second reaction is actually it is logarithmically amplified. Because in the third cycle this will be again formed like 4 copies. So it will the abundance of this will be more. So from the second cycle, after the second cycle, this will be logarithmically amplified, exponentially amplified. So this is actually a typical DNA replication fork look like. So there is a lot of difference between DNA replication, what is going inside of the cell, and how a polymerase chain reaction is taking place. So this is the trombone model of DNA replication. One replication fork. Actually, in a cell, it will be a replication fork will be like a bubble. There will be 2 replication fork will be there. So in the replication fork, there are helicase enzyme will be there, which is actually helping the double stranded DNA into single stranded DNA. Then there will be single strand binding protein will be there, which will help or which will make the single stranded DNA stable. And the primer generally used in a DNA replication process will be an RNA primer. And to synthesize the RNA primer inside the cell, there will be a primase will be there. And to remove this RNA from the replication fork, there will be an RNase will be there. So in both strands, the DNA is made not made as continuous. In one strand, it is made as continuous, and in the other strand, it is made it in a discontinuous manner. So in a replication fork, you will see leading strand and lagging strand, and generally DNA polymerase 3 is involved. And the replication fork is generally known as a trombone model, and it is bidirectional. It is semi-conservative, and there are some signal sequences are there in the DNA replication to stop it or to terminate. And after termination, there will be a recombination based process will be going on to stop the reaction and to make it into 2 copies. And if it is a linear template, if you are using, there will be a telomerase enzyme will be there, and it is actually a one-step process, and there are this is in vivo, and in the DNA replication, the whole genome DNA will be replicated, and usually you will get only 2 products. But in a PCR reaction, you are using denaturation, annealing, and elongation. Denaturation will be done by high temperature, and in the case of replication fork, even if the denaturation is happening, it is not completely becoming a single stranded DNA. It will be still somewhere it will be base paired. Only the region where it is replicating, it will become single stranded DNA. But in the case of PCR, the entire template is made it into single strand, and the high temperature will keep the DNA as single stranded. There is no single strand binding protein is involved, and the primer which is used is actually a DNA primer. And here in the case of PCR, there is no discontinuous synthesis. There is only leading synthesis is there. You cannot see any gap in the PCR product formed. And the enzyme normally used is a DNA polymerase 1. It is unidirectional only. It is not bidirectional as you see it in the case of DNA replication. It is also semi-conservative, and here the enzyme will limit the PCR product formed. Some enzyme will be able to amplify up to 30 kb. Some enzyme may not be. So the termination of DNA synthesis depends on the enzyme limit and the DNDP limit. And if it is a circular template, if you are using, you will get naked products. So when the when it meets the other end, it will not make a phosphodiester bond. So always there will be a nick will be there in the PCR product formed. So the final extension step will decide the termination or the complete process of the PCR product formed in the PCR. and there are 30 to 40 cycles is involved to get the PCR product. So it is an in vitro process. And in the case of PCR, you are actually amplifying only a specific region. But in the case of DNA replication, the whole DNA is replicated. And in the PCR, once it finished, you will get millions of copies of the specific region. So that is the difference between a DNA replication and polymerase chain reaction. So I said annealing temperature several times. So this annealing temperature is the temperature which helps the oligonucleotide to make base pairing with the template DNA. So generally, for a efficient PCR product or a PCR process, the oligonucleotide should have a Tm of 55 degrees centigrade to 72 degrees centigrade. So how you calculate Tm? This is an uh, formula what you can use. 2 into uh, A plus T. A plus T is the number of adenine and thymine nucleotides. 4 is the 4 into G plus C content. That is number of G plus C content. So you add it, that many degree centigrade is the Tm of the primer. Then how the PCR will happen in an efficient manner? Another parameters is ramp rates. Ramp rate means when you see this polymerase chain reaction, you are seeing from 95 degrees centigrade, it is cooling down to 68. Then from 68, it is going to 72. Then again, it is coming to 95 like that. So this is actually temperature. What is the time taken from a particular temperature to reach the other temperature? So this actually determines because when it is changing the temperature, the liquid inside the PCR tube will take some time to reach the block temperature. So the ramping should be in a manner that when the 95 degrees block temperature reaches to 68 degree, the liquid inside the PCR tube also should reach at a 68 degree centigrade. Otherwise, if it is quickly coming to 68 degree, it will be still at a 95 only. It will take some time to reach to 68 degrees centigrade. And if you are not giving time to set at 68 degrees centigrade, before annealing, it will go to the extension step. So you set at 68 degrees centigrade for or 55 degrees centigrade for annealing, but the tube, the liquid inside the tube not achieved that temperature. Before that, it went to 72. Before that, it went to 95. So the tube and the liquid inside the tube cannot maintain the timing what you have set it. So because of the ramping, the ramping should be ideal. Then accurate and uniform temperature, that is what the block temperature it should maintain. Suppose if you say 68 degree for 1 minute means the block should exactly maintain that. There should not be any drop in the temperature. Precise, so because of that you have to use precise temperature controller and optimization, everything. Then flexibility and design of the PCR machine, the algorithm what you have used for ramping or everything will the design of the PCR machine, everything will determine the efficiency of the PCR reaction. Some PCR machine, when you check it, you can also see a lid over it, heated lid. So that is actually helping in the evaporation loss. Initially, when we are using the PCR reaction, there was no heated lid was there, so we use mineral oil. So when we use mineral oil, you have to set a large PCR solution for the reaction. You cannot set a smaller reactions. So if you are using a heater lid, you can use a smaller volume up to 5 microliter. To 5 microliter, if you are adding a mineral oil, what will happen is when you are trying to take that PCR product, it will mix with the mineral oil and you cannot use for further processes. Even if you load it in an agarose gel, it will come out because of the presence of oil. So because of that, a smaller volume PCR can be done if you are using a PCR machine having heater lid. So this is an typical oligonucleotide what we use for a normal PCR process. So there will be a PCR primer, forward primer and reverse primer will be there. So why I am telling you this is there are some modified primers used in the PCR reaction, depending on the purpose or the goal or aim of the experiment. So the factors to be considered for designing primer is always at the 3' end there should be a GC clamp, so that the 3' end will be properly base paired with the template, so that it can do the add the nucleotide to the 3' end. So the PCR primer should be around 18 to 30 bases in length, and ideally it should have a TM of 65 to 75 degrees centigrade. And suppose when you are designing a primer, you are not able to keep a TM equal for the forward primer and the reverse primer. If you are not able to set a equal TM, the difference should not be more than 5 degrees centigrade. And when you are setting the annealing temperature in the PCR machine also, what is the lowest TM? If the forward primer is having lowest TM and or the reverse primer is having the lowest TM, what is the lowest TM in the pair? Within 5 degree, below 5 degree, you will set the annealing temperature. Suppose the lowest TM is 65 degrees centigrade, you can set an annealing temperature from 60 to 65. And in the primer, there should not be any secondary structure formation between two primers or within the primer. In a single forward primer also, there should not be any secondary structure formation. And there should not be any continuous bases like this or repeating bases like this. It should not be there. And self dimers, primer dimers should be avoided. That is, forward primer and reverse primer should not make any base pairing, or it should not make within also. If you are using a primer for cloning, we can minimal level of purification is only expected. But if you are using a primer for sequencing, you have to get a good quality primer which is purified by HPLC or by gel or whatever it is. And if you are using a mutation, for the mutation, if you are designing any primer, the mutated base what you want to introduce in the PCR product should be in the middle of the bases. And if you are using a for topocloning, should not have a phosphate modification. So this forward primer and reverse primer you have seen earlier. So this is a normal forward primer and reverse primer generally people will use. So in between, suppose if you want to quantitate the PCR product form, so in between you can add one probe that is called a Tachman probe. The 3' end there is a quencher is there, and the 5' end there is a fluorescent compound is there, which is a conjugated primer. So when the PCR is extending, so because of the 5' to 3' exonucleatory activity of the enzyme, it will remove the fluorescent part and it will be released into the solution, and the fluorescence will be accumulating. So when the cycle goes up, more number of Tachman probe will be degraded and it will be released from the quencher, so the fluorescence will accumulate. So that is why this is a modified PCR product. But this will not extend any reaction because the 3' end is blocked by a quencher. And this is actually costly. So because of that, people are also using cyber green for. So this cyber green is like ethylene bromide, what you use for gel staining. So when the PCR product double stranded DNA is accumulating, the cyber green will intercalate within the double stranded DNA. So when more PCR product is formed, the fluorescence will increase by the cyber green, and you can quantitate it. So Tachman probe and the cyber green reagent are mainly used for quantitating the PCR product. So the advantage of Tachman probe is you can have, suppose if you want to check more than one gene in a particular template, you can use a different forward primer, reverse primer, and a different Tachman probe. So you can actually do multiplexing. And this is actually MGB probe. So this MGB probe is actually, MGB is minor groove binding protein. So this will actually, the length of this primer is very short, but because of the MGB, it can easily bind to the template, and this is actually used for allelic gene discrimination. So when the reaction is going on, similarly how you have seen in the case of Tachman probe, so this reporter or the fluorescent portion will be released, and it will show the, so this is actually the portion which will, the oligonucleotide portion, which will, when it is, there is a, suppose why I said allelic discrimination is, suppose if you want to check the discrimination at this template itself at several locations, so you can make a MGB probe for this location, this location, this location, and this location, and each MGB probe will be of different fluorescent labeled. Suppose if you see here there is a binding is there, when the PCR is completing it will release, and the second primer which is to be bound here, if there is no base pairing it will not be released. So you can see the, take the reading of different reported dyes of the different MGB probe, and you can see whether that particular region is present in the gene. So that is what meant by the MGP probes. So this is actually a target gene quantification. This is also similar to or something like you have seen in the case of MGB probe, but here you are using a molecular beacon. So this also you can use for checking the mutant region or a repeat region or whatever it is which is present in the template. So this is actually scorpion primer. This scorpion primer is also used for identifying a particular DNA sequence is present in the template or not. Like that, they have the scorpion primers are used. So in the case of the difference is when you use Tachman probe or MGB probe, you also need a forward primer and reverse primer. But when you use a molecular beacon or when you are using a scorpion primer, you know there is no need of a forward primer and reverse primer. You can use with a single primer itself, you can discriminate the region. So when you do comparison of in vitro cloning and in vivo cloning, you always need a bacterial host for in vivo cloning. You need a plasmid vector or yak vector or back vector for cloning, and also you need an insert. You need a restriction enzyme. You need a ligase enzyme. Then you have to do a transformation. Then after the transformation colonies, you have to screen whether the recombinant DNA is present. Then you have to multiply that culture so that more products will be formed. Then you have to sequence and confirm that what you have cloned is of a particular DNA sequence only. And you also have to so when you get the product, your product is also contaminated with the vector sequence. So specific region will be cloned and doing it. So here there is no primer is involved, there is no DNA polymerase involved, DNTP is involved, but the cost is very higher. The time of taken for experiment will be more, and but the time of repeatability will be the time for repeatability will be more, but it will be very accurate. That is with the in vivo cloning. But in the case of PCR, we are not using any host or vector or insert or restriction enzyme or ligase. Consumables are very less. Effort is very less. Only specific region is amplified. You are using primer everything. Time for experiment is very PCR. The whole PCR can be finished in 2 hours. So the repeatability also, time for repeatability also very short. So you can do it very quickly, and you will get a. Suppose if you do a in vivo cloning, generally it will take 1 week to get the specific product to be amplified as millions of copies, but it will be again contaminated with the vector sequence. But in the case of in vitro cloning, the specific region will be amplified within in millions of copies in 2 hours. So there are different types of PCR. One is nested PCR. Nested PCR means, suppose if you have a template like this, you will design a primer for this, and it will be amplified, and there will be, in the PCR tube reaction, there will be one more set of forward primer and reverse primer within this. So this is called a nested PCR. Reverse transcription PCR means when you are using RNA as a template. So when you are using RNA as a template for PCR, that RNA has to be converted into cDNA by reverse transcription reaction. Then after that, that cDNA you are actually amplifying by the PCR reaction what we have discussed now. Quantitative PCR means we have seen Tachman probe and the cyber green. So using that we can do a quantitative PCR. Then quantitative reverse transcription PCR means again when the RNA is used as a template for quantification of the PCR product. Asymmetric PCR is the one which initially Sanger's done. That is, in the asymmetric PCR you are using only a single primer, which is generally used for a Sanger reaction. Hot start PCR means suppose if your template is having or your primer is having some kind of problem, or more site binding is there, then you can do a hot start PCR. In the hot start PCR, what they do is the Taq polymerase will be bound with an antibody. So when you start the so when you are mixing up the reactions, there won't be any background reaction. Suppose when you are using a proofreading DNA polymerase for setting up the reaction, you have to be very careful. Yeah, if the temperature is not maintained as recommended by the suppliers, what will happen is, by the time when you keep the tube in the PCR machine, all your oligonucleotide will be degraded by the exonuclease activity. So in such cases, you can use a Taq polymerase or high fidelity Taq polymerase bound with antibody can be used. So you can set up the reaction everything at a room temperature, and when you keep it in the PCR machine, because it is a hot start, the antibody will be degraded and the enzyme will be released, and the template also become single stranded. So it will further proceed for the PCR reaction what we have discussed so far. And multiplex PCR means in a single tube itself, if you are using more than one set of forward primer and reverse primer. Long range PCR means the long PCR product formed. So far, a long PCR product formed is 30 kb, but practically 20 kb is achieved, because it also depends on the template preparation. So in an ideal method, it is very difficult to get a template DNA with more than 20 kb length. So in very experimental conditions, they have amplified from 30 to 50 kb, but normally we are able to do only 20 kb in lab. Single cell PCR means if you have a very small quantity or very less number of template copies. Fast cycling PCR means like we do it in the case of quantitative real-time PCR machine. So in the fast cycling, what we will do is there won't be any annealing step. Denaturation, extension, denaturation, extension. That is called a fast cycling PCR. Methylation specific PCR means suppose if you want to do any epigenetic studies, you are using methylation specific PCR. Digital PCR means suppose if your template is mixed with more than one kind of template. When you do a normal PCR, what will happen? Which template is more in quantity? That will logarithmically will get amplified. If any slight one copy is there, the primer won't get a chance to bind to that. Or after the second cycle, from the large copy, more specific product will be formed and it will be amplified logarithmically. So in digital PCR, how they done is the dilution is done in such a way that in a bubble there will be only one template will be there, and primer and other reagent also will be there. So all templates will get a chance to get the primer and it will amplify. High fidelity PCR means there won't be any misincorporation of the nucleotides into that. And rapid amplified polymorphic DNA analysis that is actually done for identifying genetic variations. Here actually annealing temperature 37 will be used when you are using operon primers. And this another PCR that is called a rapid amplification of cDNA ends. Then in situ PCR means you will do it in tissues. Differential display PCR is done for suppose if E. coli is grown in two different conditions, there will be differential expression of genes. So between two samples, what is the gene differentially expressed? You can do PCR in two different you can do PCR reaction using the template isolated from two different conditions, and you can compare it. That is a differential display PCR. Quantitative reverse transcription PCR, it's done as a one-step process and two-step process. In one-step process means you are not setting up the reaction as a two different manner. So the reverse transcription PCR, everything will be added in the in a tube and it will continue. The two-step process means first using the RNA, a cDNA is made, then that cDNA is diluted and you will do a normal PCR. So that is why it is called a two-step process. And one more thing which I forgot to tell you before, when you are setting up a PCR reaction, the primer concentration you have to maintain 0.1 to 0.5 micromolar. DNTPC you have to maintain 20 to 200 micromolar. Taq polymerase 1 to 2.5 units. MgCl to 0.5 to 2.5 millimolar. And normally a PCR cycle you have to maintain 40 cycles. And so far practically we have achieved largest PCR product of around 30 kilobase pairs. So this is all about PCR reaction and its advancement and further use in the scientific experiments. So I will be posting videos of these different types of PCR, that is nested PCR, reverse transcription PCR. All these will be separately, will be discussing in a separate video. So thank you very much for listening to my video, and please subscribe my channel and also press the bell icon. Thank you very much. Thank you.